Begin here again, membrane structure and cell structure review. Our first objective we're going to look at is describe the structure and major function of phospholipids and the plasma membrane. So again, looking at what we already talked about, phospholipids as they were part of our fats under biomolecules. So we know that they are amphipathic molecules. We know they have a polar head and fatty acid tails. And so this makes them a perfect barrier to interact with an aqueous environment on one end and then a non-aqueous environment on the other end. So they form the perfect structure to become a barrier. And as such, they will be the most integral part of the cell membrane. All right, so let's have a look at what the cell membrane looks like. Okay, and what we're going to describe this as, this is called the fluid mosaic model. So this is another way that we refer to this, the plasma membrane, the fluid mosaic model. And what this basically means is that while we're speaking about these structures as fixed within the plasma membrane, this is a continually changing environment, right? We've got proteins being inserted, proteins being removed, phospholipids being repaired, they're damaged and renewed. We've got things moving through the membrane as well. So while we describe it as a single fixed structure, in the body, it's really not a fixed structure. We've got a lot of fluidity, there's a lot of movement, there's a lot of changes happening as far as the cell and its needs, and how that membrane has to respond to those needs. So that's why we refer to it as the fluid mosaic model. Now, as we described earlier, the most important component is going to be our phospholipids. So we'll draw those in first, right? These are going to create that polar interaction facing the inside or the outside of the cell. And then we've got the non-polar tails facing each other, okay? And so this is referred to as a phospholipid bilayer. Okay, let's put a few more here. All right, so again, we've got our phospholipid. It's got a polar head, right, which we know is hydrophilic, meaning loving water. And then it's got the non-polar tails, which we know are hydrophobic, right? Fearing of water. So hydrophobic. All right. And so this is what really forms the basis of the plasma membrane, right? So let's sort of describe that here. So the phospholipids form the plasma membrane. Okay. We know that these are amphipathic molecules. Which is what we just described here. This is called amphipathic, having dual properties with regards to water. We know that this provides fluidity and flexibility for the membrane, right? So it provides fluidity and flexibility. All right. It's also going to be impermeable to water. Again, this is what allows us to separate water compartments because water can't get through here, but also impermeable to any polar molecule. So impermeable to water and polar molecules. Okay, and we sort of described this previously as well, that if something was polar, it cannot exist in this non-polar center. Hence, it cannot go through the membrane, right? So the only molecules that can actually get through the membrane, we'll describe this a little bit going forward, but these are going to be small particles, non-polar particles, and then uncharged particles as well. And again, we'll talk about that as we go forward. Okay. Let's look at some of the other components that are in the membrane. So we've got lots of proteins in the membrane, and these can be different types. We can have what we call transmembrane proteins. So here's our large transmembrane protein. We can also have what we refer to as peripheral proteins. So here's another peripheral protein. Okay, so let's describe these. So a transmembrane protein. Let's actually draw another one here because we'll describe what this one is. All right, so transmembrane proteins are also forming an important part of the membrane. All right, what we want to say about these is that they can be transmembrane, as in they span the entire membrane, but they can also be one-sided, and that they only span 1/2 of the phospholipid bilayer, right? So these can be fully transmembrane, or they can be one-sided, right? Only on one face of that membrane. The other thing is that they are amphipathic as well. By nature of being a transmembrane protein, it also has to have that same property where it can interact with the aqueous environment and then interact with the non-aqueous environment. So by default, transmembrane proteins are also amphipathic. Okay. And then lastly is that they are permanently fixed. Okay, we'll talk about the difference between these guys and peripheral proteins, but transmembrane proteins are permanently fixed. Permanently fixed in the membrane, unless there is damage to the membrane. All right, so unless there is membrane damage, then these guys are always present in the membrane. Let's look at peripheral proteins. And forgive me, I should have colored this differently just so that we don't confuse it. So I'm going to redraw it in a different color here. Right, so we've got a peripheral protein here. And let's describe this. So a peripheral protein as the name implies, is going to be on the outer face of the cell membrane, excuse me. All right, so it's always going to be on the cytosolic side. Okay, and then this is going to easily be able to dissociate and move around. So unlike the transmembrane proteins that we described were permanently fixed, always in the same spot, unless the membrane was damaged, peripheral proteins can float around. All right, and again, this is how we refer to this as that fluid model. So these guys can dissociate and move down the membrane, up the membrane. They're always on the cytosolic side, however, but they can easily move and dissociate. All right, easily move around and dissociate. Okay. The last thing we want to say here is that these are anchored by the cytoskeletal elements. So the cytoskeletal elements, such as the integral proteins, as well as intermediate fibers, those can stick out of the cytosol and help to anchor these proteins on the cytosolic face. All right, so these guys are anchored by the, oops, the cytoskeletal elements. Okay, things like actin, etc., that are in the cell that help other proteins move around. They form the scaffolding in the cell. These are what help to anchor these proteins on the cytosolic base. Okay, so as a result, now we need to label our sides here, right? So we know that the cytosol would be on this side. Okay, and then the extracellular fluid would be on this side. Right? So this is sort of the outside of the cell, while this is the inside of the cell. Okay, and so therefore we can begin to think about the peripheral proteins as being on that inner face of the cell membrane. All right, okay, what else do we have that forms the cell membrane? We've got cholesterol. Okay, we talked about cholesterol again. It's one of these ringed fats. All right, this is kind of how cholesterol appears in this ring-like carbon structure. And these guys are going to be also important for fluidity and flexibility. So let's describe cholesterol. Okay. So by nature of being this fatty ring, what it does is it breaks up some of the watery interactions, and it basically waterproofs the membrane. It prevents water from being able to get through in a similar way that the phospholipids do, but even more so because of that very complex ring carbon structure. So cholesterol is gonna increase fluidity and flexibility. It's going to prevent leakage of the membrane. So prevent membrane leakage. Basically because it is impermeable to water. All right. And I'm going to put here waterproof. So it sort of makes the membrane even more waterproof so that things can't just sort of leak through from one side to the other, water included, right? The other thing we want to say that is that it decreases the rigidity of the membrane, and it sort of breaks up the interactions between the phospholipids as well. So the phospholipids sort of can begin to crystallize or become very rigid and fixed. And so cholesterol helps to break up those phospholipid interactions and basically decreases the rigidity we can also say here crystallization of the membrane. Right? Okay. Let's look at some of the other things we have going on. So we've got carbohydrates, and carbohydrates can be two different forms. These can be glycoproteins or glycolipids, depending on which of the membrane structures they are anchored to. So let's talk about our carbohydrates. So carbohydrates are also important in the membrane. They help with cell identification and cell tagging. Cell identification or cell tagging. This is how the cell can identify itself. It can signal other cells. If it needs to be destroyed, it can signal that via some of these carbohydrates. The other thing is that it helps to bind either lipids or proteins, right? So it's going to bind to either lipids or proteins, right? And as such, if we have a carbohydrate binding to a protein, here is this carbohydrate bound to this protein. This is going to be called a glycoprotein. All right. If we have that same carbohydrate bound to a phospholipid, this is going to be called a glycolipid. All right. So same carbohydrates, they can be bound to proteins as glycoproteins, or they can be bound to phospholipids as glycolipids. They both have a similar role. That is to signal for the cell, help the cell identify itself as one state versus the other, or tag itself either for removal, for repair, for whatever it needs to be signaling to other cells nearby. So glycolipids, glycoproteins, both formed by carbohydrates binding to some of the already present cell structures. All right. Okay. The other thing I want to just make a note of is that the transmembrane proteins are also called integral proteins. This is another term that you will see, really because these guys are so crucial to the membrane that if they are damaged or injured, the entire membrane can be disrupted, right? So they're integral to the structure of the membrane. Whereas the peripheral proteins, they can leave, they can be damaged, they can be repaired or replaced, and they will really not influence the membrane overall. Okay, I'm going to pause here just to make sure we captured everything, see if there are any questions at this point. So again, the cell membrane here is really formed by phospholipids. They are what create that amphipathic nature where we have, let's put a note of this here. We've got aqueous environments out here. So this is aqueous. This is aqueous, right? Meaning that there's lots of water. And then in the center here is a non-aqueous environment. All right. And so in order for anything to be able to get through that membrane, it has to be non-polar. It has to be able to interact in this non-aqueous environment. And so we've also got additional components that help to strengthen the membrane, but also decrease any rigidity that can be created by the excessive repeating of one structure. So if we had several phospholipids that were repeating, the membrane would become really rigid, sort of densely packed together and even begin to crystallize. And to break up those phospholipid interactions, we've got cholesterol. So cholesterol increases the fluidity, decreases the rigidity, and creates this mosaic, this moving, fluid, flexible structure instead of that densely packed repeating structure. So those are some of the features of the plasma membrane, and this is what really is able to separate what's happening inside the cell from what's happening on the outside the cell. All right. Another thing I want to say here before we move on is that because we have the non-polar center of the phospholipids, now we can begin to selectively move certain things through the membrane. So this is how we can begin to describe the cell membrane as being selectively permeable, in that some things will go through, right, depending on their physical properties or their electrical properties. Some things will be able to get through that membrane, and then other things will not be able to get through that membrane. So this is how we can begin to selectively decide what will permeate the membrane.